Bismillahirrahmanirrahim. Assalamu alaikum. Cell membrane transportation is a very important chapter for the biotechnology field. So, I'm Md Faheem Shahriar. Welcome to Sindh Biotechnologists Association. Today, I'm going to discuss about the cell membrane transportation with you in detail. So, let's get started. Here we can see the factors affecting direction of transport. So here transport is means the it can be some molecules or particle transportation from another cell to from one cell to another cell, and it usually controls in two ways. One is called passive transport, another is called active transport. Usually the passive transport, here the membrane determines the direction of a spontaneous transport. So what does it mean by spontaneous transport? It usually means the diffusion. Diffusion means that molecule goes from higher concentration to lower concentration. I can give you an example of a perfume. For the perfume, we can see that it goes from the higher concentration space to the lower one. On the other hand, we don't need any kind of external energy in the passive transportation. We have also the active transportation. Usually in the active transportation, it's totally opposite of the passive transportation, and here the energy, of course we need energy, external energy, to move the molecule against the energy gradient. It's called active transport. So it needs the external energy to move the molecule from one space to another space. We have also we are going to discuss about the driving forces acting on the molecule. It divided into two categories: chemical driving forces and electrical driving forces. So chemical driving forces, usually the concentration gradient, the direction is always down the concentration gradient. And the electrical driving forces, usually it's the membrane potential, a difference in electrical potential or voltage that usually exits across the membrane of most of the cells. So let's go to the next slide. Here we can see two pictures. Usually this one is refers to a cell, and the inner side of the cell we can see a lot of negatively charged ions, and the outer of the cell, it usually the positive ions. So here the difference means the inner portion has negatively charged ions and the outer portion has positively charged ions. This difference is called the membrane potential. So the positive and negative charges actually distributed unequally between the inside and outside of a cell. And the separation of charge is said to exist across the membrane too. When the charges are separated, a potential energy, you can also say voltage, exists. The magnitude of the membrane potential depends on the degree of charge separation. The greater difference in charge between the two sides of a membrane, the larger membrane potential. Another hand, the magnitude of electrical driving forces of an ion depends on the size of the membrane potential and the quantity of the charge carried by an ion, and it increases as either of these factors get larger. So in the next slide we can see here the electrochemical driving forces. So what does it mean by the electrochemical driving forces? Here in my previous slide I have discussed that the positively charged usually move towards the negative charged, and the high concentration of a molecule, it also move towards the low concentration space. And if we combine those together, it's called the electrochemical driving forces. So we can say it like that: the total force acting on the ions is combination of those two, I mean these chemical and electrical both driving forces. And the direction of the chemical electrochemical driving forces acting on an ion depends on the net direction of the electrical and chemical driving forces too. Here we can see the equilibrium potential. It's a hypothetical value for the membrane potential at which the electrical driving forces is equal and the opposite to the driving forces. So if you so the forces is zero here. Here comes the rate of transport. So usually the rate of transport is called flux, and the flux is the number of molecules crossing a membrane per unit time. And the diffusion, I have already told you that the high concentration molecules wants to go towards the lowest concentration. Now the most important part of this chapter, passive transport. Here we can see that simple diffusion, usually it uses biological membrane lipid bilayer. I want to discuss this one a little bit. Here, for example, this one is cell. Usually in the inner cell, the concentration of potassium ion is higher than the outer cell concentration. So there should be a leak channel is present, and through that leak channel the higher concentration potassium will go forward to the lower space, this one. And the passive transport of molecule, usually it happens, I already told that, through the membrane's lipid bilayer. And you can also read details here. Actually, for this kind of topic you have to do a self study a lot, so so that you can understand better. And in the passive transport, usually there is no need of external energy, and no energy required for this one. Here you can see a picture. There carbon dioxide and oxygen are moving towards, and by this picture you can also easily understand the topic. And the next, here is the magnitude of flux depends on the few factors, usually the concentration difference, temperature, mass of the molecule, surface area, and the lipid solubility of the molecule. Here for the concentration difference, if the concentration is greater, then the flux, net flux should be also the greater, because we have the greatest concentration, it will move towards the low concentration, and that's why the rate of the flux will automatically increase. On the other hand, we have temperature, the same one, the higher temperature. If we have higher temperature, it will also have the greater speed too. For the third one, we have the mass of the molecule. For example, if we take a larger molecule, proteins for example, and a smaller molecule, we can take glucose. Usually proteins have a greater mass rather than the glucose. So as protein has a greater mass, it will show a lower speed than the glucose. Glucose should have the speed bigger than the protein. So glucose has the larger net flux than the protein. On the other hand, we have surface area too, and lipid solubility of the molecule. This one is important. The lipid solubility of the molecule, it means most polar molecule. Usually polar molecule is the for example positively charged or negatively charged ions, or it can be amino acid, so on. So most of the polar molecule diffuse very slowly, or you can say that they do not diffuse at all, whereas usually the non-polar molecules diffuse so much rapidly or very easily there. So let's move to the next slide. Here we can see facilitated diffusion. This facilitated word is very important. It means it will take help, one molecule will take help from another molecule uh, to cross the cell. For example, here is side A and side B. There is a protein present, and here are non-polar molecule, here are also non-polar molecule. If a molecule wants to cross that place to A to B, it will take help from that protein. This one is called facilitated diffusion. And here we have the carrier is a transmembrane protein. I want to discuss about the carrier later, but I want to discuss about this picture right now. Here we can see that a protein is there attached with the cell membrane. Here the glucose attached on the on there. After that, on that time the protein feel, when the glucose is attached with this one, with the protein, on that time the protein feel uncomfortable, so it changed the direction, or it changed the conformation of this one, and move backward. You can see that it moves backward, and then it releases the glucose, and then at that time when it releases the glucose, it feels again uncomfortable. Here it feel uncomfortable, so it again change the position on conformation. So we can see that conformation and changes happening there. So in simply we can say that usually in the carrier protein that binds any molecule on the side of a membrane and transports them to the other side by means through a conformational changes. And here usually the net flux of the facilitated diffusion depends usually on the frequency of solute binding to the carrier molecule on the two sides of the membrane. So the net transport occurs from the side with greater frequency of binding. On the other hand, we can also say the rate of facilitated diffusion is determined actually by using several factors also. For example, the transportation rates, the number of carrier membranes, or the magnitude of the concentration, so on. So here we can see the facilitated diffusion. You can simply just read this one. I will focus here the carrier and the channel. There are two types of facilitated diffusion, carrier and channel. Channel is the simplest and the easiest way to describe this one. Usually in the carrier we have seen that the proteins has a conformational change, then this one will convert it and goes across the membrane. But in the channel, for example, if this one is the side of A, B, and this one the protein, and non-polar molecules go directly to that protein, in that case protein doesn't need to change its position, portion, or it doesn't need the conformational change here. Here we can see the carrier type in this picture, and this one is the channel type. Now the diffusion through the channel, here you can just simply read this one, then you can understand how the channel is proceed. Now here for the diffusion of active transport, usually in active transport, it's the uphill process or uphill across. On the other hand, the downhill process is normally the facilitated diffusion, or it means the goes from the higher concentration to lower concentration of a molecule. It's called the downhill process. On the other hand, uphill uh, occurs when the lower concentration molecule moves towards the higher concentration one. This one is called the active transportation system. Here the same goes for this one, active transportation, and here also talk about the pumping. And usually the pumping means, for example, we have a great example of sodium and potassium pumping. And sodium and potassium pump is usually responsible for maintaining the characteristic of distribution of high intercellular potassium and low intercellular sodium to their respective extracellular concentration, we can simply say there. And here primary active transport, you can watch here that the sodium ions are binding towards on the first phase, and ATP converts into the ATP. It needs the energy. Usually the ATP active transport, the ATP converts into the ATP, and the inorganic phosphate here actually the third one, we can see that the sodium is released and the potassium is also binding here, and at last potassium is got released. Now secondary transport, usually it's the primary active transport by its the use of the ion concentration here. And here we can see the flow of ions is higher concentration, high energy state, to a lower concentration, low energy state. Here on the next slide we can also see that the two kinds of direction, one is called co-transport, another is called counter-transport, or is also called anti-proton exchange. And here the transport of two substances in the same direction is called the co-transport, in the opposite direction is called the counter-transport. So let's move to the next slide. So now the cell membrane transport in biotechnology field, usually in biotechnology research or biotechnology studies, you have to read, you have a great knowledge about the cell. Otherwise you can't do the experiments, or you can't do the research for biotechnology. And biotechnology, we have the great relationship with the cell chemistry. So the references I have taken for this slide from baidu.com and from this website, and a textbook which is called Da Shui Hua Shui Shu. It's a Chinese textbook which I have translated the name for you. It's called University Chemistry by Yubin Zhang. So that's all for today. Thanks for being with me. Have a good day.